Okay, so yes, it's been a while since I've used this program in particular, but the other program had cut off all the figures. Okay, one thing, let me just check something out real quick on this. Yeah, I don't think this one. The uncool thing about this one is I can't resize anything, and I don't think I can even add. We'll see how well that works. What are we? Yes.
For the exam, you said it's a different format. It's just a few questions.
Yeah, typically. Mm-hmm.
So are they problems to work out?
It can be. It can be. It depends from chapter to chapter, the kind of things it could be.
We still have 2 hours to take it.
Well, it wasn't before in the past. You only had 50 minutes. But this year, the ones that are at night, you're going to have up to 2 hours, but it's not going to be any longer than it was back when it was just a 50-minute exam. Okay. Usually there are 5 questions. Each one's worth 20 points. But now each question may have multiple parts. It depends on what it is. Okay. So there's a hierarchy to protein structure. Okay, there's primary, secondary, tertiary, and quaternary. It's very important you know this hierarchy, okay, in the way that it goes. Primary is the simplest of all of them. And it's what you already know. So let me just get this to where it's on the same track. All right. So primary structure is the easiest in the sense that it's literally just the sequence of the amino acids. However, there's always a directionality to it. There's always a directionality to it. Okay, and so that directionality always goes from the amino terminus, which they call the N terminus, to the C terminus. Okay, so for example, if someone gives you the tripeptide ACS, that's an alanine. The amino terminus goes alanine, then a cysteine, and then what would be the C terminus? What's that amino acid? Serine. Whoops, I keep everything away. All right, so I'm going to go back here. Then, wow, that was loud. Then the secondary structure is what happens when you take that primary structure, which if you just imagine it being like a bead of pearls or whatever, like a big string of amino acids, and they start to form localized shapes. Okay, just little general localized shapes. So that's a localized structure. It's one, but now this is in 3 dimensions. Okay, now the most important part of this is that localized structure is only due to interactions with the peptide backbone. So we're not talking about any type of interaction, say for example like serine can do hydrogen bonding. Now we're talking about the R group, we're only talking about it's only due to the interactions of the peptide backbone. Okay. Let's see, there's got to be a way to... I guess every time it does that, it pauses it. So it's been forever since I've used this. They've updated this app. So let's see. This is just a review. Because I want to make sure that you get this right. Because people, I almost always ask some type of question on a quiz or an exam similar to this, and it amazes me the number of times that people get it wrong. So let's go ahead and go back to the ACS. So it's an alanine, cysteine, and serine. So if we're doing it under normal physiological conditions, the amino terminus would look like an NH3 plus. Then we have the C alpha for alanine going to its carboxylic acid. But I'm only going to draw it like that. Because what's the R group of alanine? CH3. Okay. Now, since it's forming an amide bond with the cysteine that comes next, there is no O to it. It's got to form an amide bond because it forms that bond with the nitrogen of the cysteine, okay, because that's really important. It's a amide bond. This right here is an amide bond, okay, and so that's why there is no oxygen linking the two. Otherwise, well, I mean, this won't work out. If you think back to organic chemistry too, amides would be more stable. In fact, amides are much more stable than what you'd expect. Okay. So then we have the nitrogen of the cysteine going to its C alpha to its carboxylic acid, what would be its carboxylic acid group. But now on this R group, what's the R group of a cysteine? What is it? What comes next up to the C alpha? There's a methylene, yeah, CH2, SH. There's always that linking. In fact, I'm trying to think of, off the top of my head, offhand, I can't think of anything that doesn't have a linking carbon between it and its R group. I'm trying to scan through all 20 of the amino acids quickly in my head, but because that's what happens the most. One of the common mistakes I see, say for example, is with cysteine, they put the thiol group directly attached to C alpha, or the histidine groups that they'll put the imidazole group directly attached to the C alpha instead of having that linking carbon. You know, phenylalanine has a linking carbon, tyrosine's got the linking carbon. They all have that linking carbon, and I can't think of any exceptions to that off the top of my head. Okay. Pardon? Glycine. Yeah, glycine is the only one that wouldn't. Right. Mm-hmm. Yeah, guanine is for DNA. Don't confuse me for a moment. Throw my mind for loop. Guanine does not. Okay, and so then we have, once again, you don't do the O here. That'd be a no-no, because that O is gone. It forms the amide bond with what would be the serine. So that'd be the C alpha for the serine. And then the C terminus. And under physiological conditions, you know, it's going to look like an O minus. Now we come down here to this, to the C alpha, the one for the serine. And what is its R group? CH2 OH. Now, some of the suggested problems ask you to figure up, you know, at different pHs, like what the charge is. Make sure you know how to do that. The charge, what's this charge at, assuming that we are right now at pH 7, what would be the overall charge for this? Neutral. It's neutral, because you have a +1 on one end and a -1 on the other end. All of the functional groups, the R groups that's hanging off of it, all of them are uncharged. Okay, you can get to pHs where that, for example, with the sulfur from the cysteine can lose its hydrogen. Whenever that happens, then overall it would have a -1 charge. Okay, but so you may want to practice with some of the suggested problems like that. All right, so this right here is the primary structure. It's not a secondary structure. It's literally the primary structure. It always goes from the N terminus to the C terminus whenever we do it this way, or we could have written it out, you know, ALA, CYS, SER, that's fine as well. However, once you get beyond like 3 or 4 amino acids long, they're going to start to take on a localized structure. That's what leads into the secondary structure. Oh, that worked out much better. Okay, do you have to apologize? All of the alpha helices, all of the alphas and betas, they didn't make it whenever it couldn't read it. So this is an alpha. This is a beta. Alpha helix and beta sheet. All right. So those are some examples. Examples are alpha helix for a secondary structure. Alpha helix or alpha helices, beta sheets, sometimes called beta pleated sheets. Sometimes it's called a beta strand, because you can kind of think of it like a piece of fabric. You know, it's a strand of the fabric or a strip, and then you put multiple strands together and it forms like the sheet or like a quilt or whatever. And so, same thing here. Whenever we talk about that, it'll make more sense once you see examples of a beta sheet. And it's fine with me if you drop the pleated part, because by their very definition, they have to be pleated for it to be a beta sheet. And you'll see why in a moment. It looks like corrugated cardboard or roofing material or something like that. once again, it's only talking about the interactions of the backbone and not the interactions of the R group. All right?
So we're still talking about the hierarchy to protein structure.
Right. We're going to go over all four types, but we're going to go over the first two in greater detail, then come back to the last two. Because rather than say one, two, primary, secondary, tertiary, and quaternary, we're not going to do that. Okay, because the fact that until you learn some of the parts of the secondary structure, it wouldn't make sense for you to see the tertiary. Until you learn about the tertiary, then the quaternary would make sense, or make more sense, I should say. Hopefully. All right, so in alpha helix, well, I think these are out of order. We'll find out.
That's slide number 3.
Right. Good. That's correct. I got a board on the PC. All right. So next up is what's called the Ramachandran angles. Okay. So this is what's trying to show you that amide formation that we had before, that I was talking about before. See, so here, it kind of got lost in the top. This would have been the nitrogen. There's the carbonyl group. There's a C alpha, nitrogen, carbonyl group, C alpha, so on and so forth. Okay. Now, these kind of angles, this is a different kind of angle than what you're used to thinking about. You know, in geometry, how many points does it take to make an angle whenever we talk about the angle between something? 3. You know, you're talking about, you know, from point A, point B, and point C. You know, this would be an angle. Well, that's true, and those angles are important. But in biochemistry, and also in organic chemistry, it's just that they didn't probably use this term necessarily. But there's another type of angle that's important called a dihedral angle. A dihedral angle is the apparent angle made by 4 points. So they give these dihedral angles special names in biochemistry, in peptides. Okay, there's phi and psi, and then they don't between the carbon and the nitrogen of the peptide bond. And that's because if you think back to organic chemistry, let me change colors that way. There's resonance between that oxygen and that nitrogen and that carbon. So they actually it forms what's called an amide plane, and you don't get free rotation around that bond. You do get free rotation, for the most part, around what's up here called the phi and the psi bond. Okay, that's why that's one thing that limits the structure, or limits the movement of structures in your protein. And it's also really important, too, because remember, the amide bond is the bond that was formed whenever the 2 amino acids come together. And so it actually makes it much more strong than what you would expect it to be. Otherwise, if you think back to organic chemistry, you could just have a nucleophile like water come through, smack it, it would fall apart if it was too good of a leaving group. So it's actually happier than what it would be if it was a carboxylic acid or an ester or something like that. I should say carboxylic acid, happier than it would be if it was an ester. Okay, however, where the phi and psi is, there are degrees of rotation for that, depending upon what the groups are, like what my hands would have been, if my hands were really big versus something that was really small. Does that mean? Oh. Clear drawings. There we go. All right, so the way that I want to actually, let me just, oh, that's even better. So I'm going to just draw that generic peptide backbone again. So that way you can get used to doing it over and over again. Except this time, it's not going to look quite as realistic. So that's the C, oh, whoops, that's wrong. See, it might say half a point. That's the C alpha, so there's a hydrogen hanging off of that, and an R group, which I'm going to call R1. Then it's the carbonyl, nitrogen with its hydrogen, C alpha. This one's R2. And it goes on and on and on. Okay, and I'm actually going to put in 3 here just so that way You can see it, see multiple examples. Okay, so we have this right here would be a peptide bond, so that's the amide plane, and this one's an amide plane. So on those two amide planes, they don't have free rotation. Okay, so it's limited. However, the bond, of course, this very first bond, I'm not going to count it because there's not, I mean, there are only hydrogens hanging off of that nitrogen, okay? So I'm just going to ignore that one. But this bond right here between the two carbons, between the C alpha and the carbonyl group there, that one is called a psi bond, with the Greek letter psi. Okay, then we have the amide plane, and then between the nitrogen and the C alpha, they call that one phi, or phi, depending upon your pronunciation. That's supposed to be phi. Here, let me draw it down here, that way. So the way that I remember it is psi, CC psi. Phonetically, they all start with the same letter. In reality, they don't all start with the same letter, but phonetically they do. And then the other one is phi, because I can't think of anything for it. N, C, and phi. And so I just remember CC psi, and then the other one's phi. Okay? And so likewise, if we look at this one right here that I'm pointing to, is that going to be a psi or a phi bond? It's psi. And then, of course, we have the amide, and then we have this one, which would be phi. And then the last one doesn't really count because there's nothing beyond that. Or I'm not going to count it just because of the fact that it would be between two C's, and so therefore it would become a psi bond for the next one. So that's what the Ramachandran angles. It's named after a scientist named Ramachandran because what happened was they figured up the three-dimensional structures, and for every amino acid, what are the possible phi and psi bonds? Okay? And so, based off of that, they were able to figure out every single possible phi and psi bond. And they create what's called the Ramachandran plot. And what they noticed is when they did this, amino acids typically stayed in just about 2 or 3 different spots. There were only 2 or 3 on the plot, you know. So remember, it goes from, if you think back to, was that, I don't know if they did in calculus or geometry or one of your higher math classes. Rather than saying from 0 to 360, they like to say like negative 180 to positive 180, but it's the same thing. And so this would be a positive psi versus a negative psi, negative phi and positive phi. What they noticed is that certain amino acids would always have a limited variety, which was, I tried to point that out with my arms, because whereas the phenylalanine would have been banging into that oxygen if it was at 0 degrees. Okay, and so that's where, if you notice at 0 degrees, 0, 0, whatever, then this general area, there's little to nothing there. This section up here, where you have a positive psi but a negative phi, that ultimately forms what's called beta sheets. The section down here forms right-handed alpha helices. Then we have some that can be left-handed alpha helices, and then there are other specialized, there are very few of them, but there are some other specialized secondary structures. Okay, this right here is human serum albumin, which is a protein that's very important. Where is it? Right, so it's definitely primarily alpha helical. Okay, so I went to Duke University and to Duke Medical School, and there's a husband and wife team that I had to check to see if they're still there. I don't know, they would be up in years, to say the least, because they weren't spring chickens whenever I was there almost 20 years ago. So, but their names were the Richardsons. And actually, if you've ever looked at the traditional protein structures, the ones that look like little ribbons and arrows and things like that, she is the one that actually came up with that way of showing it. She's world famous for it, and what's unusual is she actually doesn't even have a doctorate. She's an artist. She's originally was an artist. She got interested in science. I don't know if that's how she met her husband or not. She actually went on to get honorary doctorates, but she actually never went on to get a doctorate or an MD or anything like that. And so And whereas he was traditionally a structural biochemist and everything, but they lived for this. I mean, they're the ones that came up with all these different protein structures, and that's what their lab did. And so whenever I was going to go off, because for my undergraduate research, I did protein structure. I worked on a protein structure determination lab, and so it's one of the reasons why I went to Duke, because I'm wanting to work with the Richardsons because they are world famous. Like everyone knows in that field knows Richardson. Yes.
What's the difference between the red and the yellow?
It's the higher probability. So, one of the... Actually, I didn't even get to do a rotation with the Richardsons because they were winding their lab down back then. So they didn't take a lot of students on and everything like that. But I did get to meet with them, and you had them in class and got to talk with them. Their lab was just right around the corner from ours, and so I got to see them a lot. But they were hippies, no doubt about it. Okay, so... But everyone had to take a structural biochemistry class that was taught by them. In particular, she would teach, and she was this she was an elderly lady. She was kind of short and a little rotund, but she had really long hair. She always wore up almost like the Pentecostal holy buns kind of thing, But they were not Pentecostal at all. But for example, she would just like to try to demonstrate alpha helices and things. In a moment, I'll show you. She'd just take her hair down and roll it out, and like that was, or take her belt off. She'd always wear a skirt and take this belt off and do like different things. But she'd get really excited, and so we had to study all these protein structures. And she'd say, "You see this right here? This right here? That's just a little piece of sheet." And so we're all trying to, it's a big lecture hall. And we're trying to keep them. And she said, "That is a little stubborn piece of sheet right there," and all this kind of stuff. And so, but yes, but no, they had quite a lot of stories about them. But no. They are the ones, or some of the scientists that actually came up with the way the nomenclature that's used, the way that we talk about protein structure, and so on and so forth, along with Ramachandran. So I mentioned this is alpha. It just didn't come through in the... This is an alpha helix, okay? Alpha helices are really interesting. Once again, we're only talking about, and I want to point this out, notice they just put generic R groups here in this, what is this, a brown or yellow color? Those are just generic R groups. That's not, that is not the, just glycine. It could be alanines, it could be valines, something like that, okay? So we're only looking about interactions with respect to the amide backbone, that protein backbone. So if you notice here, this is a carbonyl group that's hydrogen bonding to what would be the hydrogen hanging off of the nitrogen that's right above it. And so that's what these are showing. These are showing hydrogen bonds. So we see this is what the hydrogen from the nitrogen is hydrogen bonding to the carbonyl group of the carbon. Because of this, all of the R groups stick out. And before I go any further, let me just make sure. Oh, it's a couple slides down. But because this, to me, they always look like, if it's a nice, perfect alpha helix, it looks like a Christmas tree. Because you have R groups that stick out, like a Christmas tree. And so this is just showing you some of the representations. This is the space-filling representation. But the alpha helix also has what's called a handedness. It can be either right-handed or left-handed. Okay, so if we look at this one on the far right, yeah, far right for you guys, and for me, I don't know why for you. Far right for all of us now. It wasn't like I was born with two left hands. Okay, if you notice, all of the carbonyl groups have the oxygen pointing in the same direction. Okay, so because of that, if we look at just one of these, it doesn't matter which one we look at. Actually, let me just clear this. If we look at just one of these, so this one right here, there's a dipole. Okay, so which is more electronegative, oxygen or carbon? Okay, so it means it's delta minus, and this is delta plus. Notice this would be delta minus, this would be delta minus, this would be delta minus. All that's supposed to be delta, so on and so forth. Delta minus, delta minus. And the delta pluses are all on the bottom from the way this is drawn. So because of that, there is an overall dipole moment. Helices have a dipole moment. All these little delta minuses add up. So overall, there's a dipole moment that goes up along that helix. So one end of the helix is going to have a dipole moment that's more negative than the other end of the helix. So the way to tell which hand, if this is a right-handed helix or a left-handed helix, and if you remember back to the Ramachandra plot, there are more right-handed than left-handed. So if you just had to guess, odds are. But there's a way to tell. And that's where if you take your hand, it doesn't matter which one right now, so I'm going to use my right hand, and you put your thumb in the direction of the cardinal groups in the direction of the dipole. If it's a right-handed helix, your hand will curve with the helix. See, this is coming out, and go around, coming out, as I do my pirouette. Whereas if you turn it down with your left hand, it won't. So here, there's no way. This should be, from my fingers, my thumb, sorry, my thumb be pointing up. It would have to go in, then coming out instead of coming up. Well, do that. So that's where they talk about the handedness. Your thumb is a dipole. This is right-handed because it's coming out towards you, it's going around, coming out towards you, and so on and so forth. Whereas if I try to do it this way, it wouldn't work. If I start with this carbon, my hand would be going away, then coming towards you would be the opposite. Okay. Once again, all the little squares here are alphas. So alpha helices, they just be going up and around and spiral and everything, but there are certain things that can disrupt an alpha helix. Okay. One of those is a very specific amino acid. And if you think about it, what amino acid would it make sense that could break up an alpha helix? It's proline. Because remember, proline's R group, it can't spin around at all. It's stuck. And so since it's stuck, it has limited phi side. It has very limited phi side because there is no free rotation. In fact, your body has an enzyme called racemase, which deliberately breaks it open and closes it so that way it kind of releases that whenever you have to change from one isomer to the other isomer proline. Okay. So because of that, proline creates a bend. Okay. And then, it's going to give away all the answers on this one. You can also have strong electrostatic, which is just a fancy way of saying positive and negative charges positive charges repelling with positive charges, or negative charges repelling with negative charges. So if you have two positively charged side chains right next to each other, they're going to repel. Or two negatively charged amino acids right next to each other, they're going to repel each other. And so because of that, it can cause distortion with the alpha helix. So what are some of the amino acids? What are two of the positive amino acids that could cause this charge repulsion? If they're positive, what ones could it be? Lysine. Not glycine, but lysine, in case you couldn't tell since I got it coughed off in my mouth. Lysine, and what's another one? There are two more, but there's another really big one.
Isn't the one that has a nitrogen and then two more N?
Okay, yes.
Arginine.
Arginine. And then what's the third one? Does anyone remember?
Say it again.
Oh, Consuela. Bye, Consuela. She must have thought I said something about Consuela on there. I didn't think I'd say anything that sounded like Consuela. Oops, no. That's why I needed my phone. So, because it's not a name, you're going to hear it very often. All right. That's awesome. So we have arginine, we have lysine. Does you remember what's the third amino acid that can be positively charged? It's also aromatic. There are three. Histidine. Those are three. What about the negative ones? What are the ones that are negatively charged quite often? Aspartate and glutamate. Okay, those are the ones that are the most often that are due to these charge repulsions. And then finally, we can have steric crowding. So steric crowding means they're big and bulky. They take up a lot of space. All right. And so things that can take up a lot of space are things like valine and 3-enene. Those are the branched. Isoleucine, the big branched. Phenylalanine, tyrosine, anything like that. Those are big and bulky. So at this point, I'm going to go